This is a fish. This is not a fish. Oceanic and terrestrial vertebrates share a common ancestor, which led to the evolution of conserved optical structures, like the lens, the retina, the cornea, and the optic nerve, that all allow images to be focused onto the retina and processed by the brain. Vision is used for the same things: finding food, avoiding predators, and finding a mate. So why do the eyes look so different, and why are fish so much more diverse? There are different selection pressures on underwater vision than on land. Even though light is focused in the same way as it enters the lens onto the retina, underwater vision is more difficult. The first issue is that photons are scattered as they enter the ocean. Light bounces off and is absorbed by water molecules, meaning light entering the lens and onto the retina is multidirectional, and images are blurred and difficult to focus. Light attenuation by the water molecules absorbing the blue and green wavelengths mean that the spectra is limited. The refractive index of the cornea in terrestrial vision does not limit the focusing power. However, the refractive index of the fish cornea is too similar to water and provides no extra focusing power to the lens. In summary, fish vision is so difficult as photons are scattered and absorbed, the intensity decreases with depth, as does the limitations on the spectra. The fish eye has evolved several mechanisms to cope with these. The lens is free-moving, allowing it to have both short and long-distance vision, and the lens has a refractory index gradient, an increasing density gradient from the surface to the core, so light is bent more powerfully towards the centre and prevents images being focused beyond or before the retina. The lens also corrects for chromatic aberration, where colours are focused at the wrong point on the retina, allows for long and short distance vision, and the specialised chromatic pigments in rods and cones allow for different visual environments. These selection pressures act differently with depth and habitat type, as the habitat has different compositions of scatterers and absorbers in the water, meaning a different visual environment. Let's do a habitat diversity tour. Warm water reefs are categorised by a high light intensity in the equatorial regions, and the biogenic structures create complex shadows and crevices, meaning a very complex visual environment with a greater available spectra. Prey fish, like the ambom damselfish, use vision to find green algae on contrasting coral colours, to hide from predators, and for interspecies signalling. They utilise specialised cone cells arranged in a complex mosaic for a greater range of photochromatic pigments for advanced colour vision. Nocturnal predators have to cope with a greater range in light intensities to hunt at night. Shark pupils can open and close to control the amount of light they absorb. They have lots of rod cells to allow for low light vision, and they have a tapetum lucidum, a layer of reflective mirrored crystals behind the retina to enhance vision in low light conditions. The open ocean is a less complex but more challenging visual environment. The blue light is highly attenuated and limited, and any light entering the water gets increasingly scattered with depth, meaning images are very blurred, and predators need lots of rod cells to maximise light capture in order to identify the contrasting shadows of prey above them in deeper waters. They also have their eyes positioned on the side of their heads to improve their binocular vision. When prey are very small and fast-moving, having a large overlap in fields of vision improves 3D images of prey for a moving target. This sharpens visual acuity and gives a 3D image of prey despite the open horizon. The most extreme visual environment is the deep sea, where there is little to no downwelling light, and species that undergo diurnal migrations have very variable light conditions. Most light in the deep sea is blue or green wavelengths from bioluminescence. Lanternfish undergo a great example of this migration. They use vision for detecting prey, counter-illumination, and migration. In the daytime, they are in the deep water, hiding from the tuna above. However, at night time, using their vision to monitor ambient light levels, they move upwards towards the zooplankton. They need vision to navigate the depths and to know when to return. They have lots of rods on their retinas and the tapetum lucidum to improve low light vision, and an elongated ventrotemporalis, which improves contrast with downwelling light. The barrel-eye fish uses vision to detect lanternfish above them, and by rolling their eyes with specialised muscles, they can track prey movement above them. They have specialised eye adaptations for this. The tubular green eyes have muscles to allow them to roll upwards to see through their transparent heads and locate the bioluminescent wavelengths of prey. The extreme limitations imposed on vision underwater caused a wider range of selection pressures on fish, leading to their diverse eye evolution that contributed to their increased diversity over terrestrial vertebrates.